want to welcome everybody to our next session. Very excited about this. The session's entitled Understanding the Brain, How New Technology Enhances Our Understanding of Neurological Diseases and Brain Health. So I am Dan Sem. I'm going to introduce our esteemed panel in a moment. I just want to briefly say a few words about who I am. So I'm Vice President of Business Development and co-founder of a startup company that's focused on brain health called Estrigenix. Estrigenix is a spin-out company from Marquette University, Concordia, and UW-Milwaukee, and we're focused on developing a drug for treating neurodegeneration in postmenopausal women. So we're targeting the estrogen receptor beta. I also have moved over to the dark side and into business from science, and I'm Dean of the School of Business at Concordia. I don't really think it's the dark side, but and Concordia, we have the largest MBA program in Wisconsin, and if anybody's interested, we have a special focus on healthcare innovation, so tailored to physicians and PhD scientists looking to commercialize new ideas. So, but let's talk with our panel now. So if the panelists would just sort of briefly tell us a little bit about yourself. I see we have our affiliations up here, but just a few sentences about what you do. So start with Cindy.
I'm Cindy Carlsson. I'm a physician in geriatrics and do Alzheimer's prevention research focusing on looking at spinal fluid biomarkers, neuroimaging, and cognitive changes in people at risk for Alzheimer's. And I also practice clinical medicine at the Veterans Hospital.
Thanks, Cindy. Sterling?
Good morning. I'm Sterling Johnson. I'm a neuropsychologist, and I'm in the Alzheimer's Research Center and the Alzheimer's Institute, which we'll talk about. And I study early detection of the disease. We use brain imaging and other biomarkers of the disease to understand the earliest changes that take place.
Thanks, Sterling. And Su-Chun, if you'd tell us.
Yeah, I'm Su-Chun. I'm a professor at the Department of Neuroscience and Department of Neurology at the university. I'm working on neurodegenerative disorders using human stem cells as a model system, try to figure out how the nerve cell degenerates and how to use stem cell to develop a way to deal with the neurodegenerative disorders, particularly focusing on Parkinson's disease, ALS, and so on. Outside of the academic work, I also co-founded the startup called BrainXell, and the company focuses on using human stem cells to produce nerve cells, very specialized nerve cells for building drug discovery platforms. And we are now also working on building the cell therapeutic part of the venture.
Thanks. So we have a great panel here of brain experts with bigger brains than I have, so we're going to ask some interesting questions to the experts. And I was for sure going to do questions at the end, but if you have burning questions while we're talking, feel free to raise your hand, and I think there's people with microphones circulating, so we can field questions as well. But I thought before we talk about the future and new technologies and new treatments and understanding of brain disease going forward, it'd be a good idea to start with where we are right now. So, and have a discussion regarding current treatments and technologies being used in the clinic. And Cindy is a clinician working with patients. So Cindy, I'm wondering if you could tell us a little bit about current state of the field in terms of treatments for Alzheimer's disease, dementia, brain disorders in general, and maybe where we fall short with what we have.
Yes, I'd be glad to. So again, when we go to our clinical practice, it's really different than when Dr. Johnson and we look at our research participants using novel technology. So when we go into a clinical setting, we're trying to diagnose someone based on what their symptoms are, so what they're telling us, what their family's noticing. We do some neuroimaging, for example, to make sure they haven't had a stroke or other significant brain changes. But it's really very much talking to the person and trying to establish, based on what they're telling us, what kind of symptoms they have and what the disease is. And so as we think about novel technologies, there's a huge need for better technology to be able to identify early brain changes. And as we'll talk about, a lot of the brain changes may start decades before a person develops symptoms. So in our clinical practice, we're just talking to them, seeing what kind of symptoms they have, making sure there's no other medical problems, but there's not really a blood test, a spinal fluid test, a neuroimaging test that's going to help us accurately diagnose the cause of their dementia. We do also use technology in the clinic. For example, we'll use electronic medical record and apps to try to help caregivers to some extent, but there's a lot of room we can talk about later in the panel to help us diagnose and treat and manage patients.
Right.
So with neuroimaging, we can do certain types of PET scans, so positron emission tomography scans that can help us look for certain patterns in the brain for neurodegeneration, but again, it's not specific to Alzheimer's or other types of dementias. As far as the treatments go, the treatments currently really just address that the brain has had damage and the neurons are dying, and that the neurons need, the nerve cells in the brain need improved signal conduction, so they help improve signal transmission across the synapse, but really doesn't address the underlying cause of what's causing Alzheimer's. So we can expect that people will continue to decline with the disease. So again, it's very, very much talking to the patient, not much technology in the diagnosis, and then not very specific types of therapies currently, unfortunately.
And therapies aren't getting at root cause, or more just
Exactly.
treating the cognitive symptoms, things like that.
Right.
Sterling, Su-Chun, did you want to chime in terms of where we are right now with treatments and diagnosis?
Yeah, I would just say, in terms of the big picture here, this disease affects about 1 out of 7 women over age 65 and 1 out of 10 men. And there's 5.8 million people in the U.S. right now with Alzheimer's disease. And what Dr. Carlsson just mentioned about treatment is so unsatisfying that we don't have a cure, we don't have a way to stop this disease from progressing. And that's kind of our state of affairs. And so we'll talk about, as we go along, how we're addressing that.
Right.
Yeah, it's a major issue. I think you can tell why NIH puts so much effort and the resource into it, and the public puts so much resources. And sooner or later, we will also end up with this.
Yeah.
And so we probably have to really work hard on that. I think in terms of therapeutic, it's a long way to go. And I think that's why I actually, even in my own lab, I switched to study how we get old and how we end up with that stage.
Yeah. Could you slow that down for me? Okay, so now that we're a little depressed, we sort of know the state of affairs where we are now. I think
Why we're here, right?
it's going to be more optimistic because we're going to talk from innovators about what the future holds in terms of better diagnosis, earlier diagnosis, and therapeutics. But I guess before we move on to the happier thoughts, one more thing. Probably most of you may know that there's been quite a few failures of drugs in clinical trials that have been A-beta targeting drugs. We have from Biogen, I know I'm going to mispronounce this, aducanumab, and then from Merck, Verubecestat, both failed this year. Previously, we had failures from Pfizer and Lilly and J&J and others. There's just been a whole slew of failures of drugs that have been A-beta targeting drugs, the beta amyloid plaques. So I'm wondering, starting with Su-Chun and then others chime in, what can we learn from this whole series of failures of drugs in clinical trials in terms of A-beta targeting and so on? Are there lessons to be learned? And what do you see in terms of the future? Is pharma sort of regrouping? Is there any reason to be optimistic? So I know some pharmaceutical companies are moving away from Alzheimer's because of this. So
that must be billion-dollar question, not only million-dollar question, I think. So many companies over the past 2 decades almost, right, and failed one after another. I think it goes back to the A-beta cascade hypothesis, I think, because all these therapeutics developed based on this hypothesis. That is, A-beta accumulates, and then it leads to the problem in the nerve cells, which cause neurofibrillary tangles in the nerve cells, and then leads to the deterioration of the nerve function, making the nerve connection separated like the synapse dysfunction and then neuronal death. And so that hypothesis basically says the A beta formation or accumulation is an early event. So if we can target that part by decreasing the production or blocking the aggregation or promote the clearance of A beta, it would be helpful. So that's essentially the rationale behind all these clinical trials. And there are some evidence, particularly from imaging kind of a study to show that indeed the A beta accumulation happens almost 2 decades before the patient actually get to the disease stage. And so if we block this accumulation process, certainly it should help. The problem perhaps is, to me, a couple of fold. One is when we start the therapeutics, the patient is already getting to the stage that the nerve cells are already degenerated. So you really do not have much to rescue. I think that's the stage where we target, when we target. The second part, perhaps to me, is going back to the basic science part. So is there really a cause-effect relationship between A beta accumulation and Alzheimer's disease? There is correlationship, but there is not a hard evidence that it's absolutely, there is accumulation of amyloid, and then there is a disease. And so that's probably the second issue.
Yeah.
The third issue perhaps is there are many different forms of A beta. And so which type of A beta we are targeting, So that is probably something in the future we could learn and modify therapeutics targeting different forms of A beta.
Okay.
I think there are many ways we probably can learn. I'm not an expert, but that's what I feel. That
More of an expert than I am, and maybe I'll ask Sterling and Cindy to chime in as well. But if I can sort of summarize what you just said, especially with the first 2 points, is maybe we're not targeting the disease early enough, and perhaps the beta amyloid and the amyloid plaque formation is not causative for the disease; it's just correlative. And these are maybe things that we've learned from the failures. This is really a very expensive experiment that pharma has done, and we've learned something that maybe we're not happy about what we've learned from it. So I don't know, Sterling, Cindy, do you want to add?
Yeah, I'm not ready to throw out the amyloid hypothesis yet.
I like a little controversy.
The trouble is, as Su-Chun has explained, it's such a prolonged disease. And it begins 2 decades, 20 years or more before the symptoms begin. And also, it's happening inside of a really dynamic organ. The brain can reroute itself, rewire, form new synapses, and it's really adept at taking care of itself up to a point. And then these plaques, the amyloid plaques, it's thought that they can be quite silent for this prolonged period of time, but once the tangles start forming inside of the neurons, that's when you see the correlation with cognitive decline. So it's really difficult to establish the causality of this over such a prolonged period of time, because it's longer than any of our careers probably would be, or at least take the whole arc of our careers to establish this in individual patients. But the other thing I wanted to comment on with regard to amyloid is that it's very true. It may not be the right target, even though it's the diagnostic indicator. The neuropathologists, when they look in the microscope at the brain tissue, that's what they look for to make this definitive diagnosis. But it could be, just like we saw this morning with the calcium imaging from the colon screening, you can see calcium in the aorta. The calcium is an indicator of vascular dysfunction and potential heart disease down the road, but we wouldn't want to necessarily pull out calcium and stop calcium from doing its thing in the body because it's so intrinsic and so important to that. So there's all these dynamics going into the therapy space and developing these therapies.
Right.
Lots of untold and unsolved things here.
Yeah, we'll be talking about some of that. Is there anything you would want to add? Or?
I think, so again, you're right that we haven't had any new therapies for Alzheimer's disease for 16 years. So there's only 2 groups of medicines we can use, and again, so there's a lot of room for improvement. Part of it, I think, was that in the past we used to look, again, just like I described in clinical practice, that's how they would find participants to be part of these clinical trials. They would just talk with them, do some cognitive testing, and try to guess what was going on in their brains, and then enroll them in these therapeutic trials, when really it may not be Alzheimer's disease they had. Now we have neuroimaging tools that we'll hear more about that at least can identify if there's amyloid and tau in the brains of people, but then now we're finding that, as was mentioned, you can have amyloid and tau changes in your brain but not have any symptoms. So there's really an emphasis on trying to tie together what we're seeing in the person, and Su-Chun was mentioning about what kind of manifestation does that pathologic change have in the patient. So we're trying to kind of bring those things together so we can do better studies and understand if we change the pathology decades before the symptoms start, does it make a difference in their trajectory? So that takes time. There is one study, the A4 study, that Lilly and NIH together are still looking at in people who have amyloid buildup are cognitively healthy in the 65 to 85-year-olds, and they're trying to see if anti-amyloid therapy will help change that trajectory, and that's ongoing still. So there's still a little bit of hope for the amyloid hypothesis, as Kelly mentioned.
Keep it on the table. And not to state the obvious, but for the non-experts, sort of like me, Alzheimer's disease really is about neuronal death, neurodegeneration, and then later on we see the amyloid plaques, and we really don't know what's causing the cell death, do we? So.
It's that way, but these amyloid plaques show up in our 50s, and the tangles show up 10 to 15 years later inside the neurons, and it's thought that that is what becomes toxic to the neurons.
Yeah.
Go ahead.
So we're all kind of touching on the disease is starting earlier before you're symptomatic, and so we're not testing people to see if they have neurodegeneration. Probably by the time you do see symptoms, there's already been a fair bit of cell death, I'm guessing. Right. So that kind of gets at, and you were saying, Sterling, earlier that the disease probably starts 2 decades before you start to see symptoms. So, you know, it'd be nice maybe now if we could have a little discussion about earlier diagnosis. And so maybe starting with you, Sterling, and then Cynthia and Su-Chun later, talk about what are the technologies that are being worked on right now and on the horizon for earlier diagnosis, you know, imaging, MRI, PET scans, and things of that nature. So.
Right. The wonderful advances in this field are that we can use PET scans, which is positron emission tomography, to inject a radiopharmaceutical, and it goes and finds the amyloid plaques, and it sticks to it long enough to emit these positrons that the PET scanner can pick up, and it becomes a diagnostic image that is specific to amyloid. And that's also happened for the other major protein in Alzheimer's, tau. And so with these 2 types of PET scans, the amyloid scans and the tau scans, we can now detect the major pathologies in Alzheimer's disease. And indeed, that's how we know that this disease is starting decades before the symptoms appear. And this is going to be a tool that's going to be helpful in early detection and early diagnosis. We've gotten to the point where we can look at the PET scan and we can estimate with reasonably good accuracy when that disease started and how long it's been in the brain at the time of that exam, which is critical for maybe making prognostic plans and which type of treatment we're going to do. But getting back to the treatment side, we might be able to use this technology to get the right patients into the right kind of therapy at the right time. For someone who has amyloid but doesn't yet have tau tangles, and they're asymptomatic. Maybe they need to be in a tau prevention type of a clinical trial. Or alternatively, we could maybe target amyloid therapies at that time to pull the amyloid out of the brain before it evolves into this cascade where it develops these other the other signs of AD, the tau tangles and the neurodegeneration. But we're also doing a lot of work with spinal fluid, which Dr. Carlsson has been leading.
And kind of touching on that, are there biomarkers that you're looking at from spinal fluid or wherever you're looking?
So we do collect cerebrospinal fluid from participants, and that, because it touches up directly against the brain, it gives us unique information that to this point blood tests don't, although we're working on blood-based biomarkers now. But the spinal fluid test also can measure the amyloid, the tau, but also other inflammatory markers, other markers of vascular health in the brain. So there's a variety of new biomarkers that are under investigation. I think where biotech has made the biggest difference is trying to standardize those assessments over time. So clinically, we try to standardize how the samples are collected, but having machines that are very precise, improving the precision of the measurements so they can be compared across different sites has been really groundbreaking for this area. But again, trying to move toward a blood-based biomarker is one of our big goals, but the blood has a lot of other binding proteins that confound the results so far.
And are there clinical trials in this area identifying biomarkers and this predictive capability and so on?
There's the largest is probably the Alzheimer's disease neuroimaging initiative that has combined a variety of MRI type scans, different types of PET scans, and then the spinal fluid test with cognition. So again, to see if there are these changes in the brain, what impact do they have on the person? If we have all these changes in the brain and we give someone a therapy, but they never would have developed the disease, that's not going to be helpful either. So using machine learning and artificial intelligence and other mechanisms, I know Dr. Johnson's been involved with to help us identify who is at risk for that progression, who should be in a study.
Yeah. Maybe you mentioned artificial intelligence, and we had a session earlier, in terms of diagnostic ability. Could you talk a little bit about how AI is going to help us diagnose more accurately? What I presume you're sort of feeding in the biomarkers to train an algorithm of sorts. I'm off the cuff here. I don't really know. I'm an expert. Could you explain to us? I'm not an expert. So.
Yeah, the artificial intelligence deep learning is helping us identify the synthetic markers, taking all of these things that we think are part of the disease, the plaques, the tangles, the neurodegeneration, the cognitive decline, the genetics, piecing all that together, metabolomics. We have a lot of data on individual subjects that artificial intelligence can help us with. And this project that Cindy mentioned, the Alzheimer's Disease Neuroimaging Initiative, that data is available to you. There was a question in the morning session about data availability. If you Google ADNI, A-D-N-I, you can sign up to download data, get all the genetics, all the imaging that you want, and the cognitive decline, and the labeled endpoints, including mild cognitive impairment and dementia. Mm-hmm. So there's a lot of computer scientists working in the Alzheimer's space because of this kind of study that makes the data freely available to the engineers and the computer scientists.
Right. So, and I'm kind of wondering how this is going to work in the clinic. Maybe this is a question for you, Cindy. So I'm in my mid-50s. I shouldn't reveal my age, but who cares? Yeah. So if I'm going to have Alzheimer's, it's probably beginning to happen right now, but it's not something I would get tested for. If I did a cognitive test, it probably wouldn't show up. How is it going to work in the clinic? Is it reimbursed? What's the motivation for people to do this? If there were a blood test, I could see that it's something people would be more likely to do and that sort of thing. But how do you envision this sort of playing out if the goal is to detect the disease 10, 20 years before you see symptoms?
Well, we're definitely not there yet, but how I would envision it taking place would be just as we go in for routine checkups with our primary care provider and they review our health history, our family history. I might go in and say, I'm 50 now, I have a grandma with Alzheimer's, a father with Alzheimer's. What is my risk of developing Alzheimer's? And then maybe they would do a genetic profile, maybe a blood test, maybe a brain scan, whether it be PET imaging or MRI. They may do some genetic testing and say your risk of progression is in the next 10 years is 40%. And so because of that, we're going to offer you this Alzheimer's prevention therapy that will reduce your risk. Or maybe they would say we're going to control your blood pressure better because that might reduce your risk of developing Alzheimer's or some other interventions. So I think it'll help the clinicians by, just like we do for cardiovascular disease, risk stratification, we identify what the risk factors are and try to reduce those. So hopefully we'll be moving toward that. Right now we don't have any, as we've talked about, anti-amyloid therapy or other therapies that target the disease, but we do have things we can do to probably protect the brain health without maybe changing the changes that are going on in the brain.
Okay. Yeah, and I guess that gets to the next topic, which is if you see that you're at the early stages and you were to take preventative measures, what might some of those preventative measures be? Or perhaps you would just take preventative measures without being diagnosed. You're just sort of worried about it. I'm wondering how that plays out in the clinic, or maybe over-the-counter type treatment someday as well. So whoever wants to sort of feel that.
Well, right now there's no preventive therapies. There's nothing that's been established to prevent Alzheimer's disease, but there's newer data supporting things like, as I mentioned, a new study that came out this year showing that controlling blood pressure, hypertension, has an impact on delaying onset of Alzheimer's disease. So it might be that improving blood flow, maybe it improves the A-beta clearance, or maybe it improves, the oxygenation of the brain. So there may be different factors, but controlling blood pressure, avoiding medicines that work directly opposite to the medicines we use to treat Alzheimer's, so things like Benadryl and things with what we call anticholinergic properties that might help if someone's having symptoms. Benadryl itself isn't going to cause dementia, but it could affect someone's cognitive abilities. So that, and then a lot of it comes back, we'll do these large studies that help show us that it's really exercise protects our brain against these beta amyloid changes, the high blood pressure, certain dietary factors. So a lot of things we know that are good for our health anyway may also be helpful for our brain health currently until we get the preventive therapies.
Yeah, I would just add to that that's one of the really optimistic things coming out of the prevention literature is that exercise, good sleep hygiene, good diet, good control of our health is good for our brain, and it helps the brain have the blood flow it needs and the synaptic neuronal happiness that they need to thrive. And whether or not it slows down the plaques and the tangles is something we're still working on. But for sure, it improves the overall milieu inside of the head, and the neurons thrive for a longer period of time.
Yes, I would like to improve the overall milieu inside my head. Su-Chun, you talk a lot about your interest in studying the aging process and maybe the role of oxidative stress. Do you have any thoughts on what could be done, what approaches from the prevention point of view?
I don't know. I think it all has been said. I think probably exercise. I think that's useful for everything, not just for AD.
All right, I'll do it.
And I saw, because sometime when I went home, I saw my in-law playing mahjong, the game, the Chinese game.
Yeah.
And she's over 90, and I cannot play with her because she beats me all the time. and you can tell, this kind of exercise is really, really useful for kind of delaying the cognitive decline. Another part, I think sleep probably is very, very useful. I think I have a collaborator working on sleep deprivation and sleep, how sleep helps ginger the lymphatic system from the brain so that it can clear the junks in our brain, and so it keep our brain clean, at least.
Yeah.
And I think that's probably very, very useful. And there are many other ways, definitely, but I think these are the issues I heard a lot, and it may be actually useful.
Okay. Yeah. And what role, if any, do you think oxidative stress plays in neurodegeneration?
Well, it's almost a common mechanism in degenerative disorder, particularly for neurodegenerative disorder. You look at the whole spectrum of neurodegenerative disorder from Alzheimer's to Parkinson's, ALS. So oxidative stress is involved in pretty much every aspect of the disease process. And so if there are ways to reduce or somewhat eliminate the oxidative stress part, I think that will help a lot, right?
Yeah. Well, I mean, I think a big challenge is the earlier treatment being needed and how to approach that and what are the preventative measures. Just to chime in from my own experience with the company that I'm working with, we're looking to target the estrogen receptor in women during menopause, and the Alzheimer's Society has talked about how there's a correlation between menopause and hot flashes and then later dementia. So how do you treat somebody for something that isn't going to happen for 10, 20 years? So we struggle with that because you can't do like a clinical trial for 10, 20 years. You could do a follow-on phase 4 maybe. So we've decided to focus on symptoms of menopause with a drug that will hopefully help later. But for me, that's the tricky part, is how do you justify a treatment for something that is so early stage and sort of asymptomatic? But
that's where the biomarkers are so important, if we can get good surrogate markers. And I know that a lot of Alzheimer's researchers are working with FDA to think about regulatory issues. How can we get a therapy approved based on biomarker results instead of having to wait, as you mentioned, the 10, 20 years to do a clinical trial that'll really show definitive disease results.
So then I'm just kind of curious, in that scenario, which I think would be awesome, then the clinical trial as the endpoint would be looking for a change in the biomarker levels, because it would be 10 years out before you're sort of looking for a negative result, I guess. So the endpoint is at the biomarker level, or what are you thinking?
For the A4 study is a good example. So it's asymptomatic, anti-amyloid therapy and asymptomatic Alzheimer's, so the 4 A's there. So the A4 study, what they're doing is they worked closely with the FDA before they developed the study, and the primary outcome is cognitive change over time. So they have a detailed cognitive battery that picks up subtle cognitive changes in people who are cognitively healthy at baseline, and that's the primary outcome. So that's what the FDA is going to look for drug approval.
Okay.
But they also, at the same time, collect biomarkers. They do PET scans, spinal fluid tests, other cognitive measures, other blood tests. So there'll be a lot of biomarkers, but the FDA wants to see that there's an improvement in that trajectory of cognitive change.
Sure, okay. Yeah, so on the topic of clinical trials, I know, Sterling, you've led this study called WRAP, W-R-A-P, the Wisconsin Registry for Alzheimer's Prevention. I believe you're the PI for that study. Could you tell us a little bit about it? What was the purpose and what are you learning?
Yes, it's not a clinical trial. It's a natural history, observational, longitudinal study of Wisconsinites. There's 1,500 people that have enrolled in midlife. Most of them have a parent who had clinically diagnosed Alzheimer's disease. And so these individuals who are so dedicated, they come in every 2 years, they undergo all these tortuous cognitive tests, and we ask them to do spinal taps, and we ask them to do all these brain scans, and we ask them to donate their brain at the end of their life. And
that's the best times.
Yeah, right, at the end. Yeah, after they're done using it. But they're just amazingly dedicated people, and from them, we're learning so much because they give us their histories of their sleep habits, their diet habits, their exercise routines, and with that correlated with the brain imaging, and their genetics, we're coming up with ways of understanding the early course of this disease in this risk-enriched cohort. And they provide blood every 2 years, and we've got that in the freezer, and we'll be, when these blood tests mature, we'll be doing blood tests on these individuals. And hopefully, we'll be able to put some of them in prevention trials as well when those things come along. And we also have our Alzheimer's Research Center, which also has a cohort of late middle-aged adults, and we're enrolling them with the understanding that if there's a drug trial that would be applicable for them, we'll ask them to go into it. And so it's a thriving program.
Yeah.
Dr. Carlsson leads the clinical core of our center, and it's just a multidisciplinary team involving the Waisman Center, involving computer scientists and radiologists and geriatricians and psychologists.
That's exciting. And I mean, the sense that I'm getting in our discussion is that the future for treating dementia and brain diseases is earlier diagnosis and treatment and prevention, and that's going to help a lot in that regard. Cindy, did you want to add to it from the clinician perspective? You know, because you're a big part of this study as well, right?
Right. So I think I think the combined Wisconsin Registry for Alzheimer's Prevention and the Wisconsin Alzheimer's Disease Research Center, those two cohorts, again, it really gets people before they go through those brain changes, which is what we want, is to see what's the normal progression, and then when does the disease start, and then identifying what's that earliest point where we can reach out and do an intervention.
Right.
And so there's a lot of studies that build upon the RAP study in the Alzheimer's Center, so that we can, expand and learn more. And then our center is also partnering with groups across the world, so in Australia, Germany, in Sweden, so trying to accelerate the path of discovery. So I think having these combined databases, trying to get the data out so that we can hopefully learn more things more quickly. And so that's where we like the interdisciplinary teams and having access. Some people ask for blood samples or spinal fluid samples or participants to be a part of their study, and that's what it's meant to be, as a resource so we can advance the technology and the understanding of the disease and therapies.
Great, that's great. So maybe we can move to talk a little bit about treatments and what's in the pipeline. And I know, Su-Chun, you have a company, BrainXell, that is developing a model system for aging and other things maybe. And I would imagine, I think you're looking at therapeutic potential, but you're looking at using this model system to screen for therapeutic agents. And back when I was in the pharmaceutical industry, I know that a big part of the early stages is having a really relevant assay, biological assay, before you go into animal model and before you go into people. Could you talk about the model system that you're developing and its potential for screening, and then maybe therapeutics as well for stem cells? But
Yeah. So I mentioned that I'm working on stem cells. When talking about stem cell, many of you may immediately think of, I'm using the stem cell for treating patients. But there is a very useful aspect of stem cell model is to understand how the disease process takes place. So how does it work? So it works if we take patient cells. So let's say patients with Alzheimer's disease or Parkinson's or ALS, for example. And then take a drop of blood or skin cell, and we can turn them into stem cell, the so-called induced pluripotent stem cells. And those cells can give rise to any kind of cell in our body, including nerve cells. And what we can do is to guide this stem cell to the type of nerve cells that are attacked by the particular disease. For example, the dopamine nerve cells attacked by Parkinson's disease, or the motor neurons in the spinal cord that is degenerated in ALS patients, and so on and so forth. And when you do that, and you take those nerve cells and look at them under the culture, in the culture dish under the microscope, or do whatever molecular analysis, and you can identify the cellular change and the molecular change that takes place at each stage of the processes. And by doing so, you actually kind of replay the whole pathological process from the beginning to the end when the cells die. And in this way, we will understand how the disease process of Parkinson's disease, ALS, or Alzheimer's disease actually takes place. And to me, this is very fundamental for us to really develop therapeutics, because if we know what are the key events that lead to the death of cells, and then we can find ways to block that event so that we can stop the disease process, or in some cases you can reverse it. And so that's what we do, and we did it in the laboratory with, for example, ALS. We discovered that we actually took patients' cells and we generated motor neurons, and then we found that the neurofilament tangle, the tangle in the nerve actually happened in the cultured cells. And then we built fluorescent light onto that gene, and then we use this to screen drugs in collaboration with NIH, and we find something, the compound can actually block this process. And that's, in essence, the process we are doing in the lab, and it's also doing in the company.
Okay.
And that's why the company is mostly working with pharmaceutical companies, help pharmaceutical companies to build these drug discovery platforms so that they can target their individual diseases.
Yeah.
So that's what we are doing.
Yeah, that's really exciting because it seems to me, the way you describe it, that you're getting more at the root cause, the neurodegeneration behind aging.
So that's what I mentioned at the beginning. We need to find the cause-effect relationship.
Right.
And if we identify that, that could be extremely useful. And like BrainXell, I think they did screening in collaboration with NIH. For example, we know one disease called SMA, spinal muscular atrophy, that is a fatal disease for babies. And we know exactly what is the cause, because it's the mutation that leads to the lack of production of one protein. And so we actually build these patient cells and screen compounds. And it turns out that the top 5 compounds we screened using the patient cells, 3 of them were already in phase 3 clinical trial.
Oh.
You can imagine how the
validation.
fidelity of those cells that the screening system is, if we know the exact cause.
Yeah.
And so for ALS, it's very similar. They did a screening of, I forgot how many compounds. From the FDA approved the drug library, we didn't find any compound that is effective to deal with the tangle. But we expand the screening, and we did find some compounds, and so they are developing some, designing better chemicals to deal with it. So I think that strategy is used more and more now by pharmaceutical companies.
Yeah.
And it could potentially is one way to develop therapeutics.
Yeah. So we ended on sort of started on a depressing note. This gives me a sense of optimism. I mean, a combination of what Cindy and Sterling are working on, which is earlier diagnosis and detection, and then treating the neurodegeneration, before it gets out of control, instead of what we do now, which is basically give people cognitive enhancing drugs.
Just to add to what Cindy and Sterling were working, we are actually part of their team.
Okay.
In an interdisciplinary team, because when they have the imaging diagnostic approach to identify early patients, or even not patients, basically they are early stages, and we can actually take those individuals' blood, for example, and we make stem cells out of them, and we can actually follow them over time, and to look at at this stage what the cells behave, and at the later stage when the patient indeed have the symptoms, and then what those cells behave,
Yeah.
and we can actually potentially pinpoint, what the progression of this disease process is. So yeah, it sounds like, I'm working on cells, they are working on imaging the big picture.
Connected.
The reality is we can actually work together.
Yeah.
Because
in
these
the yeah.
well-characterized participants that have imaging, cognitive tests, they also do the skin biopsies to get the cells that Su-Chun mentioned and can study.
Great, yeah. So I think we'll do questions, just one last question for the panel, and then open it up for questions. I was told we have extra time because we started late. So could we just talk briefly about the role of health tech? So telemedicine, remote monitoring of patients so they can stay in the home longer, more open electronic medical records so providers are better connected. Maybe start, Cindy, with you as a clinician working with patients. What do you see as the role for health tech the way I described it and beyond that currently and the future? So.
Right. I think one exciting step is that our Alzheimer's Center now is trying to link our participant data not only with the rich cognitive blood tests, et cetera, that we have information, but also with their electronic medical records. So we're trying to partner with our participants so that we can have access to their full health history. So that's one area that I think will help us better understand the impact of different conditions on risk for Alzheimer's. But there's a lot of work being done in trying to improve diagnostics in the home. So if somebody lives in a part of rural Wisconsin, they're not going to be able to get to a diagnostic memory center and get the care that they need. So trying to think about improving telehealth and video assessments. But also there's a great need for caregiver support. So there's a lot of different apps that are being developed to help support caregivers, connect them in with the medical health system, even more so than, and maybe I'm biased because I'm a clinician for Alzheimer's patients and their families, but the caregivers are really tied to home, tied to caring for that person for basic needs. You having to remind them to take their pills, to eat, to shower. So there's a lot of burden on caregivers. So there's a lot of work in tech, health tech, to try to improve that support of caregivers through novel technologies, computer-based, app-based. But also there's a huge safety area that tech I think could really add to. So for example, if somebody is lost while they're driving and you're calling them in a cell phone, you can track where they are, but they're not answering their phone. Is there a way that somehow that there could be a technology device that could, talk to the person and say, you need to stop driving, you need to turn around, or give them a guidance. Or if somebody falls, or if somebody only has access to a flip phone instead of a smartphone, how can we make sure they're connected and safe, that they're taking their meds properly, that they're not, again, having a fall or getting lost, things like that. So I think within the diagnostic field is huge for early diagnosis, but also the caregiver support, tying it with the medical records so that the other healthcare system members know, oh, they're not managing their diabetes well, that's because they have dementia, not remembering to take their therapy, but also tying it into other safety issues that could make a huge impact on the patient and their families.
Yeah. And I would have to think it ties in with AI and some of the tools you're talking about.
Absolutely. There's some really talented people working on this at the university, and I'm sure across the state. But yes, in engineering here at UW, there's people working with sensor technology and in computer science. We're using AI to help us improve brain imaging for Alzheimer's disease, similar to what Dr. Burnside mentioned earlier today for breast cancer. So I think there's just wonderful opportunities to bring AI into the health tech platforms.
Great, yeah. I wish we had more time for this topic, but I want to open it up for questions, so we have time for some questions. And there's people with microphones.
At the beginning, you started discussing recent failures of therapies and even discussed how some groups are sort of stepping away from this area. With regards to this, what challenges do you face or foresee facing when trying to convince others to provide the resources necessary for the discovery, development, and testing of new therapies, and how do you address these challenges?
Others in terms of like funding agencies, but also investors and things like that?
Yes.
Okay. Anybody want to field that? NIH is funding this, I would imagine.
NIH is funding it, so.
I think part of the issue is working with the pharma companies to get the right patients into the right treatments at the right time. And I think that has not happened well enough yet. The failures that we've seen recently have been mostly in the symptomatic phase of the disease, where the brain has already failed, so to speak. It's already got dementia, And we need to, that's a tall order to bring a brain back from that place. So we need to be in the prevention space. And I think as pharma gets more into that philosophy, I think there'll be another generation of clinical trials.
I think probably is evidence-based result. You know, if you show strong evidence that the potential treatment may pan out, and farmers are still interested. You know, even though over the past decade or more, most farmers were kind of left the neuroscience field because treating neurological or psychiatric is really tough. So much investment, so little reward.
Right.
But in let's say just recent couple of years, so many farmers jump into the cell therapy field, for example, pulling dollars for, let's say, cell transplantation therapy for Parkinson's. You see how much dollar put it nowadays, almost $1 billion. Just for one disease. And so because there is strong evidence from animal studies, even including monkey study, to show that there is efficacy. And I think if we come up with some evidence to demonstrate that it may work, and I think probably will convince the farmers or some other industry to come back. At least at the moment, even though we talked so much about Alzheimer's, there are many other neurological diseases that you can target. And at least NIH puts huge resources for Alzheimer's disease research. I'm actually still optimistic that something will come out.
And your company, BrainXell, is
Yeah,
kind of banking on this.
we are working hard on it. We actually just launched the cell therapy for Parkinson's. We are producing GMP-grade cells, and hopefully some people will say, I'm going to put dollar in your company. And we started with Parkinson's disease, but we also studied with other diseases that have no treatment, let's say spinal cord injury, stroke. And so for spinal cord injury, for example, we actually generated very specialized cells to target the spasm, pain, and motor function. And then we showed them it worked in mice and rats, and then we move on to monkeys to demonstrate that it will work if we put them together. And so hopefully that will convince people that it's worthwhile to pursue that venture.
Yeah, and I remember a few years ago all the reports about companies dropping their CNS programs.
Yeah.
But I'm starting to see a resurgence. Lundbeck has always been interested, and Takeda is looking to grow their program. So I think we're coming out of the cold freeze, I hope. Other questions?
Hi, I was wondering, so it sounds like one of the keys to getting to an effective therapeutic is finding an early diagnosis. And I was just wondering if there are any like longitudinal studies utilizing individuals with with Down syndrome or familiar Alzheimer's, maybe looking for inflammatory changes in their blood or other biomarkers, since their onset of disease is so much earlier and more rapid, and if you think what the efficacy of such a study might be and how applicable it might be to the broader population. And then a second question I had about the therapeutics that have recently failed, do you know if their failure was in the ability to clear the amyloid plaques or simply to restore cognitive function? I guess what I'm driving at is if there were an earlier diagnosis, is it possible that these therapeutics that we've already developed might have been effective?
That's a great question. I think the answer to your last question is these therapies do clear the plaques out of the brain. They do it very well, but to your point, they have intervened too late and have not been able to bring cognition back to where it needed to be.
That's where I think the A4 study will be so helpful in that if people, or before they have symptoms and they clear the amyloid and it still doesn't help, then that's going to really show us that it's not helpful, because that's probably an early enough target. But then for the Down syndrome group, there's a study, it's called NIAD, N-I-A-D. What does that stand for? I forgot. But it's a multi-site study for people with Down syndrome, because as you alluded to, that people with Down syndrome are at much higher risk of developing Alzheimer's disease. Most of them will progress to develop some of the amyloid changes that we see in Alzheimer's, but at a much earlier and faster rate. And so they are doing, it's almost like the Alzheimer's disease neuroimaging initiative, where they follow people over time. They do cognitive tests, spinal fluid tests, brain imaging, so that we can better understand and hopefully use that as a model too to help people with sporadic Alzheimer's as well as people with Down syndrome and their risk for Alzheimer's. So that's a great study. It's being done here at UW and the Waisman Center as well.
Great.
Yeah, the Down syndrome model is a terrific model to look at the longitudinal issue of AD. I think I remember that in 2012 I initiated this program with a few of us here, tried to apply for the NIH award to really combine imaging, metabolomic stem cell model to look at how the imaging correlates to the cellular change. And I think now, even though at that time it was not regarded as a good model, now I think NIH started looking at this. And I think Waisman Center, where I am, is we have a registry of Down's patients, and we are working on this part.
Okay.
It's the highest enrolling of the NIAID sites. But the concept is that we look for rates of change in people with Downs, people with autosomal dominant Alzheimer's disease, and people who are preclinical. We can see what their rates of change is on these biomarkers and cognitive tests, and use that to design the next set of clinical trials to alter that rate of change in the pre-symptomatic frame.
Right. Thanks. Do we have any other questions?
Your panel discussed exercise and blood pressure for early detection and prevention of these brain diseases. So my partner's research explored the effect of lutein, which is the nutrient in leafy greens, on early stage neural differentiation. I was wondering, have you observed or seen any kind of nutritional factors play a demonstrable effect on possibly early stage detection or even prevention?
Haven't seen any.
There's a lot of studies looking at different nutrients. There's been studies looking at vitamin C, vitamin E, a lot of the components of curcumin. So there's a lot of studies that are looking at some of these antioxidant effects impact, but I haven't seen any that have shown definitively, if that's your question, about definitively any changes in biomarkers that look like they're going to affect cognition as well.
Mm-hmm. But
it's being studied though.
Okay. All right. Well, I think we're at time, so it's been great talking with this panel of experts, and let's thank our panel for a great discussion. So.